Hi, in this video, I'll be talking about the techniques used to study cognition and behavior, using accuracy and reaction time measures, various neuroimaging techniques, and some of their strengths and weaknesses, as well as single-cell recording and lesion studies. One of the basic ways psychologists use to study cognition is to observe cognitive responses such as accuracy. For example, you could conduct a study to see how accurately someone can recall words in two conditions, those that are related, such as apple and fruit, and those that are unrelated, such as coffee and house. How accurately we can recall words in these two conditions can sometimes reveal how these words are represented in memory, and how they are encoded and retrieved. Similarly, in my last video I talked about a study done by Yeagerland Todd and colleagues, and how older participants were more likely to interpret facial expressions given to them through pictures as fear, while younger participants were more likely to interpret them as shock or sadness. How accurately they respond to these pictures and correctly interpret them as fear will reveal how far along their frontal lobes are from becoming fully formed. Another basic measure psychologists use is reaction time. The study of reaction time, how long a participant takes to complete a cognitive task, is known as mental chronometry. For instance, similar to the accuracy example, how fast a participant will respond to saying apple after the word fruit compared to the word house. This can, again, sometimes reveal how these words are represented and stored in memory. Based on these differences in reaction time, it's important to note that researchers will take both accuracy and response time measures, because in these types of studies, participants will sometimes make a speed-accuracy trade-off. They'll sacrifice accuracy for reaction time, or vice versa. Sometimes, techniques differ in what is called spatial and temporal resolutions. Spatial resolution in this sense basically means how a technique looks at location, so how it samples a small or large area, from a single cell to part of the prefrontal cortex, for example. Temporal resolution, on the other hand, is the same idea, except it samples time in milliseconds or several seconds. Okay, so the first technique is eye tracking. Eye tracking studies, as the name suggests, is a type of research that looks at eye position and movement while performing cognitive tasks. For example, when in a reading task, a participant will read a sentence and might find a word to be ambiguous in meaning, and their eye movements will be tracked as they go back to that word in an attempt to decipher its true meaning in the context of that sentence. This type of study is also used in facial and scene recognition studies. An advantage of eye tracking techniques is that it has good spatial and temporal resolutions, and is less invasive and inexpensive compared to the past. Nowadays, the accuracy and speed of eye tracking methods is much better. With the advancement of technology, we can pin down eye movements to a single pixel on a computer monitor, and measure them accordingly from pixel to pixel. And where it used to be head clamps to hold us steady during the test, nowadays there's no such thing, again, thanks to the advancement of technology. However, one disadvantage is how we interpret eye tracking information can be subjective. For example, in facial recognition eye tracking, if someone looks at a particular area of the face more than others, it doesn't necessarily mean it has any form of significance in the given task. The second technique is computer models, in other words, the use of artificial intelligence. This is interesting because using computer models, cognitive psychologists can simulate processes which can then be used to test theories of cognition. So if we want a computer to simulate a specific task, all we have to do is input the algorithm necessary to do so, which allows for an easier and quicker way to test these theories. Next, I'll talk about methods of linking cognitive processes to neural structures. One of the methods is single-cell recording. Some Nobel-winning research was conducted in the 60s to map out the visual system in animals using single-cell recording, where microelectrodes were inserted extracellularly into single cells, sampling multiple cells in different areas of the brain. The aim was to map out the visual cortex and to see the response properties of all the different cells involved. One of the questions the researcher would ask is what factors, what independent variables can cause changes to the behavior of these cells, such as moving stimuli or brightness intensity, because there are cells that only respond to movement and those that only respond to brightness, color, and shape. This is a good example of research with excellent spatial resolution, conducting research at a cellular level. This next technique I've mentioned in my first video, in the introduction to Cognition and Neuropsychology, and its Lesion Techniques. This is another classic technique where researchers damage an area of an animal's brain in order to discover and find out its functions. So what is normally done is, an animal will perform a task, then knocked out, the desired brain area damaged, and woken back up, and will then undergo the same task. For example, can an animal still go through a maze and navigate themselves out of one if their hippocampus was damaged? If you haven't watched my second video, the central nervous system, one of the responsibilities of the hippocampus is the creation of new memories, which in this case is required to ensure, to the best of their abilities, that the animal doesn't end up in the same area of the maze more than once. Although lesion techniques today are much better, in the past, attempting such techniques were quite unreliable. because trying to damage a specific area of the brain might have resulted in damage to other neighboring areas. But modern techniques are now more accurate, as we'll find out shortly. Now we'll move on to techniques used to study brain injury. First of all, there are multiple causes for brain injury, from strokes to physical and mental traumas. And of course, it's assumed that if an area of the brain is injured, its function is likely affected, and we are interested in finding out these effects to better understand cognitive processes. So, these techniques include single and double disassociations. Single disassociations involve designing a study where participants with brain damage are compared with those without brain damage when completing certain cognitive tasks. And single disassociations are found when participants perform normally on one task but poorly on the other. An example would be a study done by Chalice and Warrington, who got patient A and patient B to perform on two memory tasks, one short-term and one long-term. It was found that patient B performed well on both tasks, while patient A performed well on the second long-term memory task, but poorly on the first short-term memory task. Patient A was the one with the brain injury, and this is what single disassociation is. This shows that the damage patient A had affected his short-term memory, among other potential effects to his cognitive functioning. Although, a factor to consider is the difficulty of these tasks. For it to be a fair experiment, both tasks have to have equal difficulty, which will typically be up to the researcher to decide and make sure of that. Double disassociations, on the other hand, involves designing a study with brain-damaged participants performing two different tasks, such that participant A might perform well on task 1 but not task 2, and participant B might perform well on task 2 but not task 1. This can show which brain areas are affected by their respective brain damages, accounting for the problem of task difficulty affecting the results in single disassociations. Lastly, I'll talk about neuroimaging techniques. I mentioned earlier that there are more modern sophisticated lesion techniques that provide a better, more accurate way of looking at how a damaged brain region affects its functioning. This is called transcranial magnetic stimulation, and this involves using a tool in order to temporarily disrupt the functioning of a brain area, any brain area, before performing a cognitive task to see what is affected. So very similar to the old lesion techniques, but much safer and far less likely to have any significant long-term damage. And obviously, these impaired tasks are done after they undergo the same task but with a healthy brain. This is another good example of research with good spatial and temporal resolutions that is non-invasive. Moving on to electroencephalography, this involves using electrodes on the scalp to measure electrical signals in the brain. And if we average all these electrical signals over multiple trials on the same experiment, we can get what is called event-related potentials, which is basically electrical activity in the brain, which is a response to physical stimuli, an association with mental activity, and in preparation of action, so it's a measured brain response. This technique has good temporal resolution and is inexpensive but has poor spatial resolution, even when using over 100 electrodes. Another problem is what's called the inverse problem. Using this technique, we can only view what's on the surface of the brain, and therefore we can't find out where these electrical signals come from deeper in the brain. Another technique is magnetoencephalography, which involves the use of a machine to pick up magnetic fields generated by the brain, because the brain's electrical currents generate magnetic fields which are then picked up by the machine. And while performing cognitive tasks, we can look at which areas of the brain are affected by these tasks. This too has good temporal resolution, but poor spatial resolution and is invasive. Next is positron emission tomography, which I'm sure a lot of you have heard at least once in your life. This measures regional cerebral blood flow in the brain by injecting radioactive water into participants while performing cognitive tasks. Blood which contains this water flows into activated brain areas, and an image of the brain is then generated to see which areas exactly are activated. This is a technique that has good spatial resolution, very good actually, but poor temporal resolution and is very invasive. Lastly, I'll talk about functional magnetic resonance imaging. This is basically like PET, as it measures blood flow in activated brain areas, with blood containing oxygen which gives out magnetic fields picked up by the machine, which then gives out an image of which areas of the brain are activated in certain cognitive tasks. Like PETs, it has good spatial resolution, but poor temporal resolution. It's invasive and very expensive. So, that's it for this video. Next, I'll be covering memory. Thanks for watching, and I'll see you then.